Okay, good afternoon, everyone, and a very welcome to the third webinar in our monthly series, Unraveling T-cell Recognition, Insights from Immunology and AI. My name is Hashem Kui, and I am delighted to be chairing today's session. So as you know, this series takes place on first Monday of each month, where we explore cutting-edge discussion and perspectives at the intersection of T-cell immunology, AI, and machine learning. Our goal is really to foster an open and inclusive community and discussion for anyone interested in T-cell immunity across academia, industry, and beyond. So announcements are made about the seminars and speakers via our LinkedIn account or the dedicated mailing list that we have created for this. So please stay tuned for that. And one thing to note here is that actually our initial intention was to have just 3 seminars for the theme data, opportunities, and challenges, meaning that today to be the last one. But considering the importance of this theme and also considering that we have had a number of very interesting studies have published very recently, we have decided to continue it in at least 2 or 3 more seminars in this topic. Therefore, please stay tuned. And I think actually next week I will have a surprise for you. And last but not least, in terms of housekeeping, as you know, we will be having our conference at the end of September about the same topic. So I am very delighted to welcome many of you in that conference and hackathon. So it's my great pleasure to have Dr. Wanlu Liu here. She will be talking about exploring human T cells and TCR repertoires at single cell level. And just a brief bio about her. Dr. Wanlu Liu is a tenured associate professor and assistant dean at the Zhejiang University, University of Edinburgh Joint Institute, Zhejiang University. She also serves as an honorary lecturer at the University of Edinburgh and as an adjunct researcher at the Department of Rheumatology and Immunology at the second affiliated hospital of Zhejiang University. Dr. Liu earned her Bachelor of Medicine from Zhejiang University and her PhD from UCLA. Her research lies really at the intersection of bioinformatics and computational immunology, with a particular focus on developing databases and AI-driven algorithms for single-cell and spatial omics. Her work aims to uncover the phenotypic and functional diversity of T-cell receptors. She has published in leading journals such as Cell, Nature Methods, Nature Communications, Nucleic Acid Research. Dr. Liu leads projects funded by the National Science Foundation of China and is an active member of the Immune Cell Biology branch of the Chinese Society for Cell Biology, as well as a committee member of Zhejiang Bioinformatics Society. It's a great pleasure, Wanlu, to have you here, and thank you so much for sharing your data and your work with us. So please, the stage is yours.
Thank you, Hashem. Can you guys hear me clearly?
Yeah.
Okay, perfect. Yeah, thanks a lot for the invitation and for the kind introduction. So it's my great honor today to share you guys about like the T cells and TCR stories recently published from our group. So I'm currently like principal investigators in Zhejiang University. And to start with, I think within this group, I don't need to say too much about T cells. So basically, T cells play a very vital role in adaptive immune responses. For example, in the anti-tumor responses, T cells is able to recognize like the PMHC peptide presented by the ATCO tumor cells, and then the activated T cells can then expand it and then recognize the tumor cells and kill it. So to have a T cell that is in a good function, the effector function is mostly mediated by the gene expression of those cytotoxic enzymes, chemokine receptors or inhibitor receptors. And the antigen-specific recognition is mediated by the TCR PMHC complex, which is like one of the major focus or research interests in this group, I believe. And TCRs are extremely diverse due to this well-known VDJ recombinations happens in the DNA. So basically, during VDJ recombination, TCR or BCR, they will be able to randomly choose like one V gene and D gene and J genes, and then to form a TCR alpha beta heterodimers. So the gene, along with the gene recombination, junctional diversity, and chain recombination, so it's theoretically estimated it can be up to 10 to the power 15th of the TCR diversities. So this TCR and BCR diversities basically enable the recognition of virtually unlimited antigens. And it's very important. So how one of the key questions that is happening in the field is like, how can we identify functional TCR? The reason to identify functional TCR is very important because TCRs, it can recognize those wide range of low-level or intracellular antigens, and FDA have approved like 2 TCR-based therapies in the recent years. And however, like the long-lasting questions in the field is like, how can we identify functional TCR target within almost like the infinite diversities? So in the previous years, when like the next generation sequencing technique have evolved, so scientists basically use this TCR BCR seq. So we will collect those bulk immune like cells, and then we extract the mRNA and cDNAs, and then we will use like a set of primers that is specifically for TCR or BCR, and then we will do the sequencing with NGS and do the bioinformatic analysis. So the good thing about bulk TCR and BCR-seq is that it can give us this VDJ gene usage and this CDR sequence, which is the region that is directly recognized the antigen peptide. And it can also give us a full length of individual alpha chain or beta chains for TCR or light chain or heavy chains for BCR. However, it's very hard for the immunologists to perform like downstream validations using the bulk TCR results is because, first of all, there's no cell identity because this is like a bulk sequencing. So like the gene expression of those cells is unknown. And very importantly, like there's no receptor pairing information. Like we may get like hundreds of very enriched alpha chains or beta chains, but we don't know how they pairs. So it's very hard for downstream functional validations. But like T cell phenotype is defined by both gene expression and TCR. So as we mentioned before, like in T cells, there is a very heterogeneous gene expression states. Like the T cells can have this naive or like states, and they can be induced into cytotoxic functional states or can go into like this pre-dysfunctional exhausted states. And on the other hand, like TCR is very highly diverse. So it's very important to capture both like the gene expression and the TCR information at the same time in order to help us better uncover those functional TCRs. With the development of the single cell technologies, the single cell immune profiling technologies have been developed. So the core concept for this single cell immune profiling actually is quite simple. So just it's just a regular like a single cell RNA-seq library preparation strategy by doing single cell separation and mRNA extraction, and then we will make a single cell RNA-seq library. At the same time, we will use TCR or BCR specific primers to amplify those immune receptors. And in the end, what we will get is like a gene expression profile for each of the cells. And at the same time, we will have a cell barcode linking those gene expression profile to the TCR alpha chain, beta chains, or BCR light chain, heavy chains at the same time. So basically at the single cell level, we will be able to capture the cell state information and the antigen receptor information at the same time. So actually, like capturing like the T cell transcriptome or gene expression and the TCR information have long been like tried in the field, like starting from 2016, like using multiplex PCR and single sequencings, and like around 2016 to 2013, like scientists have used fax-based T cell sorting and then sequencing to couple like the T cell state and the TCR information. And in 2014, like from Mark Davis' lab, they have developed this NGS sequencing of the TCR pairing and the transcriptome at the same time. And then with the development of the drop-seq and really make like the single cell technology enter into this commercialized, and in 2018, like 10x Genomics have made this single cell immune profiling technique a commercially available kit. So it's really entered these big data areas. And with the massive data available in the field, it really started to call the need for the computational or system approaches to understand those data or analyze those data in more depth. So the first thing our lab did is we established a human antigen receptor database, which is called HUARDB. So what we did is like, this is published in 2022 in Nucleic Acid Research. So what we do is at that time, we collected all the publicly available single cell immune profiling data sets, and then we use a unified like pre-processing like pipeline to like process all those data, and then we collect and get the gene expression matrix and the TCR or BCR sequence and VDJ gene usage for each cells. And then we did like the T cell, B cell subtype prediction along with the clone type definition. And we then like curated all those information into a cloud database. So in the first version of our database, in total, we collected like about 400,000 TB cells in total. And among them, like I would say 90% of them is T cell informations. And in this database, this is an online like web-based database. So what the user can do is you can look into each individual data and then like check the gene expression or the cell subtypes that you're interested in. And at the same time, if you click on each of those T cells in the database, it will also tells you the T cell receptor information, including like the VJ gene usage, and also like very importantly, like the CDR3 amino acids, and which may allow like the downstream validations for immunologists. So in the first version of our database, we collected about 400,000 T cells with both its transcriptome and TCR alpha beta information. And along the past several years, our lab is continually like collecting publicly available single cell immune profiling datasets. And last year, our database have enriched to more than 2 million T cells collected in this database. So the database, the data we have collected now, like this is a million scale pan disease single cell gene expression TCR alpha beta data collection. So it's collected from more than 70 studies and more than 1,000 samples and 500 individuals, and spanning like different disease types. So what is contained in this data are, first of all, very importantly, like the disease meta information, like the individual ID, the disease type, like whether it's solid tumor, inflammation, autoimmune, etc., and also the tissue type, like whether it's like tumor infiltrate lymphocyte or PBMC or lymph node or other like tissues. And at the same time, we use this software called ARC-HLA to infer the HLA genotypes from the single cell transcriptome information for each of the individuals. And importantly, we have this for each of the 2 million T cells collected in this data. We have its gene expression along with its TCR alpha beta information. And this is the data overview. So you can see that about one third of those data is collected from solid tumor, spanning like different kinds of tumors, and also have quite a few data coming from like inflammation or autoimmune, and also infections. We also collected some other like conditions like healthy controls, longevity, fragility, or IRAE, which this is immune-related adverse effect from the PD-1 or other checkpoint blockade therapies. So after we collected this huge collection of human T cells information, the first thing we were wondering is that whether we can integrate those T cells together and do some data mining and analysis. But first challenges we face is that this for this Atlas level single-cell RNA-seq integration is it has very, very large batch effects because, as I mentioned before, all those data are collected from different publications, more than 80 labs probably, and the cell number is huge. So we are integrating more than millions of cells. So using the traditional PCLU map strategy, you can see that the batch effect is very large. So basically, those cells, they will just cluster by which data set or which publication comes from, instead of the cell type it belongs to. As you can see that the right UMAP is colored by its cell types. It's basically the same T cell types is scattered across the places. So that's why we start to develop single-cell RNA-seq integration methods based on deep learning, specifically designed for Atlas level integrations. So we use this variational autoencoder deep learning frameworks for single-cell integration. And the reason for VAE is because previously, several methods such as SCVI, SCANVI, SC-poly, or SCALEX have been built upon VAE used for single-cell integrations. And the effect for VAE-based single-cell integration is quite good. So so that's why we also developed a variational autoencoder-based framework for this task. But the differences for our framework is, first, we use a batch invariant encoder. So the core ideas for variational autoencoder-based integration is that we input some gene expression matrix, and we fit it into this variational autoencoder framework, and then it learns some latent representation in the hidden space. And what we want to do is we want to use the parameters learned from this latent representation to reconstruct my gene expression matrix. And if my reconstructed gene expression matrix is very similar to my input gene expression matrix, then I will assume that my latent representation is learning the key features that is needed for this task. So in this VAE framework, our methods we adopted a batch invariant encoder because we are dealing with hundreds of batches, and this design also allows our methods to do very fast transfer in learning, which I'll show later. And then instead, our method incorporate the batch encoder in the latent representation. So which means that our decoder is a batch variant decoder. So during the batch encoder, we will also, other than the study name, we also allow the user to input the atlas name. So which means that if you have different T cell atlas or other atlas with contain similar cell types, you can also use our methods to do cross-atlas integrations. And another unique thing that we designed is this cell type predictors from the latent representation. So we added multiple cell type category prediction module here. So this design allows us to perform cross-atlas cell type alignment and cell type annotations. So the key functionality of our SC Atlas VA is, first, it can be used to build reference atlas. Basically, we have many CDA T cell datasets, and we can do cross-study integration and get to a reference atlas. So this is the basic function. So as I mentioned before, because of our model design, it also allows us to do cross-atlas integration with cell type alignment. So if we have like several atlas that contain all T cells, we can do subtype alignment and like different atlas have different cell type annotations. So you can align to see how those cell types, even though with different names, they can be aligned. And most importantly, it can allows for transferring learning of the query datasets. So if we got a new CD8 like single cell datasets, it can very easily to map those T cells into our reference maps and then do automatic cell type annotation. So in our in the first trial, so after we have this data ready and after we have this like deep learning model ready, the first like demo we did is we like constructed a human CD8 T cell reference atlas. So basically, we focused on the CD8 T cells that is collected from our 2 million like data sets, and then we those data is collected from like all human individuals and in different conditions like infection, inflammation, IRAE, solid tumor like leukemia, etc. And those T cells, CD8 T cells, they coming from like circulating or tissue or TILs. So because this is not only solid tumor, it also have like those inflammations. So we also have like T cells from the tissues. And each of those T cells in this collection have the single cell gene expression along with this paired like TCR alpha and beta informations. And in the downstream, we also perform some population analysis like the transcriptome analysis and TCR expansion or TCR sharing analysis. So in the first task that we demoed, our model is we built a pan-disease CD8 T cell atlas. So as you can see that after we integrate all those like 1 million CD8 T cell together, we manually annotated like 18 CD8 T cell subtypes, including like the naive T cells, like the central memory and the resident memory, cycling, exhaustion, or like T cells recently activated based on those like microgenes that is known before. And we have checked the expression of those microgenes in those 18 T cell subtypes that we annotated. And from this UMAP, you can, I hope you can appreciate that the batch effect is nicely removed with our SC-Atlas VAE, like even by integrating like more than 60 studies, this our method can correct those batch effect very nicely. And we look into like the UMAP, the human CDA reference map that we get. And the first thing that we notice very interesting is that the CD8 T cells coming from the circulating and coming from the tissue or TILs, they separate into two parts on the UMAP automatically, and which is along with it's like the disease sources. For example, like in the tissue or TILs, mostly it's coming from solid tumor, while like in the circulating, mostly it's coming from like infection, etc. And we also looked at like the 18 CD8 T cell subtypes enrichment in different tissues. And you can appreciate that in the TIL, like the tumor infiltrating lymphocyte, mostly they are enriched for exhausted T cells and effector T cells, while in circulating, we have most of those like naive T cells or memory T cells, and in the tissues we have those tissue resident memory T cells, validating like our cell type annotations. And we also look into like the cell type composition in different disease, and you can see that those exhausted T cells, they are like the proportion of those exhausted T cells, they start to increase in those solid tumors. And because every individual T cells in our million, like this human CDA reference map, have the TCR information. So we also did some repertoire features of those those T cells. So we first calculate the D50, which is measuring the TCR repertoire diversity, and you can see that in the naive T cell, like the TCR diversity is really high, and along with the differentiation of the T cells into, for example, the memory or exhausted T cells, like the TCR diversity is decreasing, and basically the T cell is clonally expanding. And this is also like along with the disease conditions. So you can see that the D50 of each individuals in our data sets and comparing, for example, healthy with solid tumor, like the TCR diversity in solid tumor or other like leukemia or other disease conditions, they are significantly reduced. And we can also like see like the between different T cell subtypes, like what's the clonal sharing between those T cell subtypes. And you can see that there's very dynamic like T cell clonal type like sharings or dynamic transitions between different T cell subtypes. So we look into like the exhausted T cells, and we only extracted those exhausted T cells from our collection, and then we like did the unsupervised clusterings, and we get three like exhausted T cell subtype, which is we named as GZMK-TX, IDGAE-TX, and XBP1-TX. So these three like T cell exhaustion cell subtype, they have a very unique features. For example, the GZMK, they are like have a gold term enriched for INF gamma or IL-1 or TNF, and also have those chemokines like CCL genes upregulated along with the GZMK upregulation. For ITGAE, it's like the upregulation of those cytokine production and type 1 INF production, and also have those like natural cell killer genes like KLR, KIR upregulations. For XBP1-TX, is have this like response to ER stress with the upregulation of XBP1. So when we look into like what's unique about those three different exhausted T cells, we immediately realize that these three different exhaustion T cell subtypes, they have very different clonal sharing patterns with other cell types. As you can see from this map, which is plotting the TCR clonal sharings of those three exhaustion T cell subtypes with other T cell types. And you can see that for ITGAE-TX and XBP1-TX, they have like one of the major like cell types that is clonally sharing with them. For example, ITGAE-TX is mostly sharing with ITGAE-TRM, and XBP1-TX is mostly sharing with the ZNF683 T factors, while GZMK-TX have more like a diverse like sharing with other cell types. And we also look into like some representative clonal types. And for example, in this clonal type 1, like which is for GZMK-TX, you can see that it have the clonal sharing with ITGB2 TRM and like the ITGAE-TX. While for the ITGAE-TX, it's mostly like sharing between ITGAE-TX and ITGAE-TRM, while like the XBP1 is mostly sharing between these two cell types. So while like we are analyzing those exhausted T cell in our collections, and we realized that there are actually like several previously published like pan-cancer T cell atlas. As I mentioned before, our collection is a pan disease, so it's not only focused on cancer. So in these two papers, one is published in Science, which we name as T cell landscapes. In their collections, they collected like from 21 cancer types, and they among those like more than 300,000 T cells they collected, there are about 100,000 CD8 T cells with both transcriptome and TCR-seq. In another study published in Nature Medicines, and they also collected like the single cell T cell data from more than 16 cancer types. And among those 300,000 T cells they collected, there are about 200,000 CD8 T cells have this transcriptome and TCR-seq. So because of the unique cross-atlas integration design of our SC-Atlas VAE, so we demoed the cross-atlas integration and alignment function in using SC-Atlas VAE. So what we can do is we can integrate like the T cells from two atlas, for example, HUARDB, which is our like collection along with the T cell landscape and the T cell map. And you can see that this is the upper UMAP. This is the cell type annotation from our like CD8 T cell atlas, and this is the cell type annotation from the T cell landscape. And we can also do the same thing for HARDB and T cell maps. And you can immediately appreciate that in the T cell maps, they have a lot of the cells that is missing. One of the reason that we think is because in this two pan-cancer T cell atlas, most of those data are collecting from TILs. So which means like that they might miss a lot of those T cell states that is in the circulating state. And along with the cross cell type alignment function module in SC-Atlas VAE, because different atlas usually tend to have different cell type annotation. So with this cross atlas alignment function, you can easily align like the cell type annotated in one atlas with the cell type annotated in another atlas and see how consistent or different they are. And this is an example of like the T cell landscape annotation and our annotation. And you can see that most of them are like quite consistent, while there's some like slightly difference. So when we like take a deeper look into the exhaustion CD8 T cell subtypes in our data sets, along with the T cell landscape, we realize one thing is that we annotated 3 exhausted T cell subtypes, and in the T cell landscape, they annotated 4 exhausted T cell subtypes, and the alignment between our atlas and the T cell landscape actually is not very consistent. You can see that the cell type, for example, the ITGAE TX we annotated, part of them is this ISG and part of them is this 6CL13 like TX they annotated. So this bring us to annotate a higher resolution of exhausted T cells. Actually, this is requested by the reviewer, like asking us like why we are missing some of the exhausted T cell subtypes. And when we after like we basically take those exhausted T cells in our data sets and then take a deeper look and then like annotate a high resolution map of like our CD8 T cell atlas. And now we're comparing like our high resolution TX annotation with the original annotation from the T cell landscapes. And currently like those T cell types, for example, the ISG TX subtypes now can be like very nicely aligned with our GZMK TX ISG like T cell subtypes. And the marker genes between the two like the T cell landscape TX and our landscape, they are very, very consistent. So another thing that draw our attention is this IRAE. This is an immune-related adverse events, which is which usually happens like after the PD-1 or CTLA-4 like blockade therapy, which is like those activated T cells or proliferated T cells. They might enter those tissues, which is not only in the tumor cells and may recognize some autoantigens and because of the cross-reactivities and cause some other like autoimmune like symptom, for example, the arthritis or colitis. So in our data sets, we actually have this type of data, which is caused by like the CPI colitis. So this drives us to ask the question is that whether those CPI colitis, like driven by the PD-1 blockage therapies, is it like the cell type or the phenotype of the T cells is similar to those autoimmune disease like UC, or it's different? So we basically extracted those healthy clone or like UC or CPI colitis along with some solid tumor TIL, which is treatment naive T cells, and then we extracted those cells and then did some more in-depth analysis. So what we do is we extracted those cells and then we look into those clonal expanded T cells, which means their TCR is expanded more than 3. And then we look into like what cell type are there. And you can see that in the healthy clonal, mostly is this ITGAE, tissue resident memory cells, while in the UC, in the autoimmune like colitis, it's mostly this ITGB2 TRM. But in the colitis, in the CPI colitis, it's mostly ITGAE TX, which means that in those CPI colitis, instead of those inflammation, instead of likely caused by those tissue resident memory T cells, it's very likely to caused by the exhausted T cells. And this phenotype is very similar to the solid tumors in CRC used as a control. And we, in our data collection, we also have this CPI colitis. So we also see very similar phenotype that it's an exhausted T cells that is mostly clonally expanded and enriched. Another like analysis, another cell subtype that caught our attention is this IOTCK, which is innate like T cell killers. This novel cell type is defined in this 2 papers. And they are like alpha beta T cells with TCRs, and it's identified in both human and mouse, and it has very highly cytotoxic potential, and especially in the tumor localized innate like CD8 T cell subtype. And it's characterized by the high expression of TCRαβ and FCER1G, so these 2 marker genes. So the reason why we notice this cell type is that when we're doing this integration of our dataset collection, along with the T cell landscape and T cell map, we realize that most of those like T cell type that captured in our data sets align with the 2 T cell types very nicely. But actually in our data sets, we collected like one unique cell subtype that does not exist or captured in the 2 other like T cell atlas. So this cell type, we name it as IOTCK-like cells. The reason why we call it IOTCK-like cell instead of IOTCK is because that the original IOTCK identified in the Li Ming's group's paper, they actually identify in the peripheral, in the tissues or TILs, but actually we identify those cells in the peripheral. So those IOTCK-like cells, they show high expression of the IOTCK marker gene, which is FCER1G and TCRαβ. And very importantly, it shows very highly like cytotoxicity, especially in the IRAE. So which means they might be like functional relevant in IRAE or solid tumors. And when we look at like what T cells, what other cell type sharing like clone type with IOTCK-like cells, and we identify those like TAMRAs or memory cells or effector cells. But something caught our attention is that we find that the mate cell, which is the invariant CD8 T cells, like they use the invariant T cell alpha chains, they actually share a lot with this IOTCK-like cells we defined. So in the beginning, we were wondering like whether this is because of like we misclassified the cell type of the mate cells. So we extract the mate cell and IOTCK cells out, only this 2 cell type, and then redid analysis. And we find that this IOTCK markers is like highly expressed in those IOTCK and also some of the mate populations, and they also express those mate populations. So which means that the cell type annotation for mate and IOTCK is not problematic. And most importantly, you can see that the part of those IOTCK-like cells highlighted here, they are actually using those invariant like AJ genes of the mate cells. So which means like they are very likely to be either RTCK using the mate TCR or mate cells that is having a different phenotype. That is the state, the cell type, the cell transcriptome is very similar to those RTCK cells. So because as I mentioned before, because we use the batch invariant variational autoencoder data structure, this allows us to do this zero-shot transfer very fast. So what does this mean is that we don't need to when we have like a new set of data, like for example in our paper we collected like another like 10 single cell RNA single cell RNA-seq for T cells, either with TCR or not, like about 500,000 of those T cells from pan cancers. And we don't need to retrain our SC Atlas VAE because we can directly take out those embeddings or those weight trained by SC Atlas VAE and then project those query cells onto this human CD8 reference map that we constructed. And we can get those like marker gene heat map along with like our reference. And it can also project into those were the query cells that we collected, and it can calculate the odds ratio of those cell composition. And you can see that in those PBMCs, mostly it's enriched for TEMOS, while in the TILs, it's mostly enriched for like the TX and TRMs. And our methods can also directly project those query cells onto our reference map and then do the automatic cell type annotation along with like the reference annotation we have, and can look at the cell type, the TCR column sharings between different T cell subtypes. For example, the two different type of exhaustion T cells, the ITJE-TX and GZMK-TX. We also confirmed that the ITJE-TX in the query dataset is mostly coming from ITJE-TRM, while GZMK-TX have more like a diverse range of cell sourcing. And the last validation we did is for the mate and the ILTCK. So whether those ILTCK cells, they are using mate TCR or they are mate cells that having ILTCK like transcriptome. So we look into those published like mate cells sorted with the MR1 tetramer. So those were mate cells with known like antigen specificities. And we transfer those data from this published data into our reference map, and we can identify like a proportion of those mate cells that is actually expressing both the FcER1G and TRBP. So which means those are mate cells, but they may exhibit different like phenotypic diversities. And this is also confirmed from another like MR1 tetramer sorted mate cell single cell RNA-seq, and from their study, they actually in their original UMAP, you can see they identify like part of those this like cells clusters that they did not know what it is before. And when we checked the gene expression for this two marker gene, and you can immediately realize this like mate classes, they were actually highly expressing this FcER1G and TRBP. So we think that it's more likely that it's the mate cells that is having like the phenotypic diversities, and part of them is exhibit the ILTCK phenotype. So in summary, like we have curated a database which is called HUARDB, and this is a link to our database. You can explore it online. This is a database for human single cell immune profiling, and mostly currently mostly is 90%, more than 90% of the data is single cell TCR-seq data. And we also curated a reference for pan disease medium scale human CD8 T cell reference atlas. And we have developed a deep learning-based tool, which is called SC-Atlas VAE. This is a method for Atlas-level single-cell RNA-seq data integration and automatic T-cell subtype annotations. And from our study, we also generate some insights, like we identify 3 T-cell exhaustion subtype with distinct clonal sharing pattern, and we find that the diverse T-cell subtype contribution in autoimmune inflammation versus this IRAE inflammation. And we also detect this ILTCK-like cells in the variables. So in the end, I would like to thank my 2 super talented graduate student who is like the first author of the database and also the Nature Methods paper, and my collaborators and fundings. And thanks for the invitation.
Thank you so much, Wanlu. It was really great. Very impressive, both in terms of generating data or collecting data from different studies, data processing, checking the quality of the data, and also developing these very nice methods to help us understand the data. So we have time for questions now. Please either raise your hand or write your questions in Q&A or in chat so that I can see and read them for Wanlu to answer. Yeah, I can perhaps start with one question. In your model development for dealing with batch effect, your variational autoencoder, as far as I could see, you used only gene expression and you didn't consider T-cell receptors. So that actually makes me to ask how this batch effect method, which is at Atlas level, is comparable to other widely used Atlas level batch effect that they just take gene expression and do the job.
Yeah. I think this is a good question. So, first of all, like I think if we are going to find functional TCRs, integrating like both the transcriptome and T-cell information is very important. So this is something that we are working on, like developing the multimodality models to integrate those information. And the second question is like, what's the difference of our SC-Atlas VAE versus like previous like SCVI and etc. So first of all, like we have had like the cross-Atlas like integration and comparison alignment, cell type alignment module implemented in our algorithms. And very importantly, like because we are developing this for Atlas level integration. So if your task is designed for like very large batches, like what we are working with, it's not just like several batches. So if you are working with hundreds of batches or like very large data sets, so maybe SC-Atlas VAE is a better choice. Yeah.
Okay, excellent. And then one question from Busan, and he's asking, actually, he has a number of questions. The very first one is that how did you select or benchmark the HGV choice?
Yeah.
And since this heavily influenced integration, do you have a set framework you advise?
How did you select or benchmark the HVG choice? Okay, so actually I didn't, because of the time limit, I didn't like talk about the benchmarking in this presentation, but actually in the paper, if you go to see that, we actually spend a lot of efforts doing different kinds of benchmarking. So how to choose the HVGs and how to do those hyperparameter searches, like how many layers of those hidden, like of those models are all tested. So you can go to check the paper in more details. And can SC-Atlas VAE handle dataset integration across compositional distinct samples, PBMC integration with CD8 T cells? Definitely yes. So, well, in the beginning, like we designed SC-Atlas VAE mostly only for like the integration of the T cells. But as Patrick mentioned, this is just a single cell integration tools, so it can be used to integrate like different type of cells. But it might need to fine tune or to to be more careful about the data or about the parameters searches. Can SC Atlas very extended Atlas example, for example, within the framework of I guess this is to set different to projection in that unseen cells that are not lost using the SC approach. I'm not sure what what this question is about. To
Yeah,
adding more adding more data sets or
yeah, sorry, at the beginning I should have basically add this clarification here by HGV. He meant highly variable genes for those perhaps not familiar with this framework. The other question he's asking is that can single cell atlas variational autoencoder handle dataset integration across compositionally distinct samples, for example, PBMC integration with CD8 T cells?
yeah, the answer is yes, but even though like we initially designed this for the integration of T cells, CD8 T cells, so it definitely can be used for other purpose. As Hashem mentioned, this is just like a single cell RNA integration tools. Yeah.
And the third question, can single cell atlas variational encoder extend atlases, for example, within the frameworks of single cell ARCS or ScanV, or I guess this is slightly different to projection in that unseen cell types are not lost using the single cell ARC approach?
Yeah, actually, I'm not sure exactly what does this question means. Is regarding about adding more data that has a new cell type. Okay, so projection often forces the transfer. Yeah. So this is an excellent question. Yes. So if you have more data that if you have like a new cell types and definitely you should not use the zero shot transfer and you have to retrain the SC Atlas via yourself and then probably you because as you mentioned, if this is a new cell type, you probably have to do the annotation yourself. Yeah, so what we are doing here is hoping to provide the reason for the zero shot transferring is we think like we have collected like a wide range of CD8 T cells that should have covered like most of the CD8 T cell subtype. So we created like a very convenient tools for the field or immunologists or people don't have like too much computational background can automatically annotate the CD8 T cells. Yeah.
Okay, and another question, which is more kind of a technical question, because here, when you are facing with this atlas level data, and when it comes to TCR repertoire analysis, basically how you define the uniqueness of TCR, because those come from different samples, different individuals, different studies, different tissues, and so on and so forth. So how you define the uniqueness of each TCR and how you define the clonotype?
Yeah. This is a great question, and I think this question only come from the TCR expert like you. Yeah. So how we because the data set we collected it has the individual information. So as I mentioned before, those more than like 2 million T cells is collected from more than 580 like individuals. So we define like the clonotype based on individual.
Okay, so yeah,
Yeah,
okay.
so definitely we detected like a handful of public TCR that is have exactly same CDR3 alpha beta but shared across individuals. So this is public TCR. We have observed like more than 3,000 public TCR clone type like this. Yeah, and as like reported before, a lot of those public TCR are known to recognize common viruses like CMV, EBV, or influenza, etc.
Yeah, and another question is that basically, again, this question is because your QC and your batch effect correction pretty much were all on gene expression, but later on you were answering, a number of repertoire level questions. So sometimes there are some inconsistency between QC if you go gene expression way to if you go TCR repertoire level. I mean, sometimes you see a very nice T cells that they have passed the QC from gene expression, but perhaps you don't have alpha or beta chain information, or the the information for them were very poor. So how do you deal with that?
Yeah, that's a great question. So actually, all the 2 million T cells collected in our data sets are those what we call high confidence T cells. So it must have a transcriptome that pass a QC and also a paired TCR-alpha beta pass a QC. So it's only like offering chance of alpha or beta. So these cells will be filtered out. So actually, if we didn't do such stringent filtering, we should have like way more T cells collected.
Okay, I don't see any more question. If that is the case, I would like to thank you again. I would like to thank everyone on board today. And yeah, please join us again 2nd of June. And I promise that we will have actually a surprise speaker. Okay, thank you.
Thank you.
Have a
Thank
good
you,
day. Bye-bye.
guys.
Thank you.
Yeah, bye-bye.